Good afternoon, everyone, and welcome to another webinar sponsored by the North American Vascular Biology Organization. I'm Enar Cuervo. I'm from the University of Illinois at Chicago and a member of the NAVBO Education Committee. I will be moderating today's session. We are pleased to welcome our speaker, Hyung Chun. He's an associate professor of medicine and pathology at the Yale School of Medicine. He will present his research entitled Endothelial Regulation of Metabolic Homeostasis. I would also like to welcome Catherine Clapham, also from the Yale School of Medicine. She will monitor today's questions. How we handle questions is in two ways. Throughout the presentation, you can type your questions into the question box in the control panel. These questions will be answered at the end of the presentation. So Dr. Clapham will compile them and then pose the questions to Dr. Chun once we're done with the presentation. After we're done with posing these questions, at the end of this question and answer period, provided that there's time, attendees will be able to ask additional questions live in real time. So if you still have a question, please raise your hand by clicking on the hand icon on the left side of your control panel. You will be recognized. I will mention your name, your mic will be unmuted, and you will be then able to ask your question live. If you experience any technical problems, please click the Help tab at the control panel, then scroll to the bottom of the Help screen for the GoToWebinar technical support phone number. We remind you that this webinar is going to be recorded and archived on the NAVBO website for future use. Now we have Dr. Chun today. He received his bachelor's degree at Harvard College and his medical degree at Johns Hopkins University School of Medicine, after which he pursued training in internal medicine and cardiovascular medicine at Stanford University. His research group focuses on endothelial mechanisms that regulate vascular homeostasis in health and disease. He focuses on disease contexts such as pulmonary arterial hypertension, atherosclerosis, and heart failure. Today's webinar will be closely related to his paper in Science Translational Medicine. Endothelial apelin receptor regulates tissue fatty acid uptake and is essential for apelin's glucose-lowering effects. You can find the link to this paper in the handout section of your GoToWebinar control panel. With this, I'd like to welcome Dr. Chun.
Great, thanks so much, Anar, for the introduction, and thank you, Bernadette, Anita, for arranging me to present today. Also want to thank Vinicio and Katie Rose Clapham for moderating down the road. But it's really a pleasure to be here. Thank you all who's listening now and possibly in the future for being here as well. And I'll go ahead and get started. So I would just start with a brief overview. So, as most of you, or almost all of you know, diabetes has a significant risk for cardiovascular complications, and wanted to kind of share with you some of the background clinical information in terms of how we treat diabetes and what the limitations are in terms of how that affects the cardiovascular complications associated with that disease. And wanted to share with you some mechanisms with respect to how the endothelial cells might be playing a role in perhaps both of these contexts, is a metabolic target as well as a kind of vascular homeostasis target, and focusing predominantly on the apelin, role of apelin signaling, which many of you may be familiar with. And lastly, share with you some thoughts on how we think we might be able to push this towards something that we can potentially test in humans in the human disease context. So I'm a cardiologist by training, and we oftentimes see patients who are diabetic and who also have cardiovascular disease. As pretty much all of you would know, cardiovascular disease, there's a significantly increased risk of cardiovascular disease in patients who have diabetes. These patients get many more cardiovascular interventions. They have many more myocardial infarctions and strokes, and a significantly greater chance of dying from heart disease, cardiovascular disease than non-diabetics. Certainly, so this link has been validated for over the years now. The challenge has been that then the thought would be that when you treat the diabetes, and predominantly I'm talking about type 2 diabetes, once the diabetes is treated aggressively with aggressive glucose reduction and control, one might expect that that would lead to decrease in cardiovascular disease outcomes. Unfortunately, as many of the studies listed here have shown over the years, aggressive treating of the hyperglycemia in diabetes has not really consistently led to improvement in the macrovascular outcomes. Now, there have been a number of studies that suggest that microvascular disease do improve with aggressive treatment of hyperglycemia, but macrovascular disease such as coronary artery disease and stroke have not really been shown to be affected significantly by treating hyperglycemia. And that's represented in just a couple slides I want to share with you from some of these large clinical trials, where 2 trials published about 10 years ago now show that there's really no real benefit to intensive aggressive glucose control in the cardiovascular outcomes of patients with diabetes. What one would hope that intensive control would have a significant improvement, but unfortunately none of these ended up panning out to have a significant improvement in cardiovascular outcomes. So then the question, and this is kind of highlighting the findings of these papers, that there really is no reduction in the macrovascular cardiovascular outcomes in patients who undergo aggressive treatment of their hyperglycemia in diabetes. We can fast forward a few years later, and there have been now some promise that actually some modalities of treating the hyperglycemia in diabetes may have a benefit. One of those drugs is empagliflozin, which was, the study came out about 3-4 years ago now, demonstrating that this particular drug is an SGLT2 inhibitor, may actually does provide benefits from cardiovascular outcome. And this was death from cardiovascular disease was significantly improved. Also, death from any cause was also significantly improved. The latest line of thinking now is that this may actually prevent heart failure and heart failure related illnesses. So there are a number of trials ongoing that that's directly looking at the role of this drug and this target in heart failure outcomes in large-scale clinical trials. Second agent is GLP-1 analogs also have demonstrated some trend towards improving cardiovascular outcomes as well. These are 2 agents, one called semaglutide and liraglutide, both of which showed promising data that there is perhaps specific drugs that are used to treat the hyperglycemia in diabetes, which may provide some degree of benefit from a cardiovascular perspective. So with that as a background, that although over the years most of the drugs have really failed to show cardiovascular outcome except these some newer agents, the question that kind of based on our research over the years is, could this apelin pathway, a GPCR signaling pathway, potentially represent a new line of therapeutic target that can potentially dually improve the hyperglycemia and also reduce the cardiovascular complications in type 2 diabetes? And this is a signaling pathway that my lab has been working on over the years. And even when I was a trainee, I started looking at this pathway while I was working with Tom Katermos at Stanford. And I just want to highlight some of the findings that I won't have time to go into for the sake of today's webinar. But it's an apelin APJ GPCR pathway has been implicated in cardiovascular development, not just by our group, but by others, many others across the world. Where it has both a role in the cardiac development as well as in the vascular development. There have been a number of systemic vascular disease processes, some of which I'll touch upon a little bit later, where this pathway seems to be implicated in atherosclerosis as well as other mechanisms of endothelial dysfunction. And again, topic I won't have time to go into. We've spent quite a bit of effort in looking at the role of this pathway in the context of pulmonary vascular disease, including pulmonary hypertension. And there's suggestion, even in human studies, that this pathway might have a role in human disease, human pulmonary arterial hypertension, but we'll save that for another time. So the receptor for this pathway was originally called APJ, and then AGTRL1 or angiotensin receptor like 1. The latest official name is apelin receptor, APLNR. First cloned about 25 years ago using homologous screening to the angiotensin 2 receptor type 1. It is a fairly typical 7 transmembrane GPCR that has a reasonably high homology to the angiotensin 2 receptor type 1. Expression is high in the heart and the lungs. Lungs has the highest expression, as well as the vasculature, and so many studies have suggested it may be more venous specific than arterial, than expressed in the arterial cells. But there is data that arterial endothelial cells may express low levels of APJ, whereas predominantly the highest expression seems to be on the venule side and perhaps some capillaries. It's similar to angiotensin receptor, but does not bind to angiotensin 2 itself. And it has a number of downstream activation targets, including the G-alpha I protein. Until about a few years ago, I would have called apelin the only ligand, but now there are two ligands. Apelin is one of the two ligands, and the second one is called elabella. So apelin has been around for many more years, so it's been studied more extensively. It is generated as a pre-pro protein that's 77 amino acids long, that's cleaved into smaller active isoforms. And there is a cleavage that occurs at the C terminus by angiotensin converting enzyme type 2, which was thought to render it ineffective. But I think there's some debate as to what is really an active apelin versus what is an inactive one. There's also high expression of this ligand in the heart, lungs, as well as the vasculature. And there have been many studies looking at the effects of this recombinant apelin peptide in different models of animal, including demonstration that this, when you infuse the peptide, there's a significant increase in cardiac inotropy. These are some cardiac MR studies showing that the cardiac contractility is improved with apelin. There's been a number of studies demonstrating that acute infusion of this peptide leads to fairly reproducible but rather transient decrease in blood pressure. This is one example of just one of the many studies showing this. And we have seen this in our hands too. There is a short-lived reduction in the blood pressure, which seems to return to baseline fairly quickly. And a number of studies by Ann Eichmann and others have shown that this actually has a major role in angiogenesis as well. So, it has a number of downstream effects, and some of the mechanisms for these effects are listed here. Again, it's been studied fairly extensively, so this is not a complete list of all the mechanisms, but some of the earliest mechanisms are listed here, including the vasodilatation, which seems to be mediated strongly by eNOS activation, as well as angiogenesis with its link to VEGF in different contexts of vascular angiogenesis. And there's some evidence that VEGF might reversely target apelin and APJ's pathway by inducing APJ expression. And I personally got involved in this pathway as a postdoctoral fellow, where we were initially interested in how this pathway, through the receptor APJ, might interact with angiotensin receptor type 1. And there has been a number of studies suggesting that these GPCRs can actually form heterodimers with one another, and I was curious to know if that might also be the case in the case of APJ and AT1R. And we confirmed that through immunoprecipitation that these two receptors can form heterodimer, as shown here. We confirmed that by FRET analysis that APJ and AT1R can form a heterodimer. We show that perhaps that some degree of this dimerization might be modulated by the different ligands that are present, perhaps more dimerization in the presence of angiotensin 2, with perhaps apelin not having an issue, not having any effect on that. And we also showed that when you stimulate with angiotensin 2 in conjunction with apelin, addition of exogenous apelin can inhibit the angiotensin 2 mediated downstream signaling target. For this study, we use a luciferase assay showing that ANG2 induced NF-kappa B activation, AP1, SRE, and NFET activation was significantly downregulated in the presence of concurrent administration of apelin. So there seems to be some receptor dependent regulation of angiotensin II signaling by the presence of the apelin signaling and apelin receptor. So we wanted to figure out, could potentially apelin affect angiotensin II function in vivo? So we carried out a mouse athero study. Alan Doherty had published previously a very nice model of ApoE knockout mice on high-fat diet and chronic infusion of angiotensin II, demonstrating a developing severe atherosclerosis as well as aortic aneurysm that oftentimes can lead to rupture. So we wanted to see, and then there was just a very accelerated model of atherosclerosis where these mice develop atherosclerosis within 4 weeks' time. So given kind of our premise that perhaps apelin antagonism of ANG2 might be a relevant mechanism in vivo as well, we sought to test this mechanism in vivo in the ANG2 athero model by using these 5 groups. Group without any infusion, group that received angiotensin II only, group that received angiotensin II plus apelin infusion, and this angiotensin II plus hydralazine was more or less a blood pressure control that a reviewer had asked for, and lastly, angiotensin II plus apelin plus LNAME, again, a different type of blood pressure control where the blood pressure was higher in these animals. And what we found was that in the animals that received angiotensin II only, we had a significantly increased athero as expected based on Alan Doherty's previous studies. When these animals received combination of apelin and ANG2, the athero was significantly reduced, and modulating the blood pressure with hydralazine had really no effect. And then this last context is LNAME to really block the eNOS effect. Had minimized the effect of apelin itself, and we confirmed a similar finding in the apelin knockout mouse, where the apelin ApoE double knockout had greater degree of athero compared to the ApoE control mice. And all these changes appear to be fairly independent of the lipid profile, which was not significantly different among the different groups of animals, as shown here, and the blood pressure. Here is just a showing of the blood pressure. So now this study is quite old, so I don't want to dwell on it too much, but just to point out the fact that there appears to be a dimerization between AT1, angiotensin II type 1 receptor, and APJ, which appears to have a modulatory effect on the AT1 pathway. In addition to the pathways that are targeted by APJ itself, including potential role on cardiac contractility, eNOS activation, as well as working in synergy with VEGF to induce angiogenesis. But so these studies were some of the initial studies that I was involved in to look at the downstream mechanisms of apelin as related to other GPCR pathways, as well as some of its own intrinsic activities downstream. And then just to highlight the fact that it does seem to have a vascular homeostatic effect where there was a significant reduction of atherosclerosis. So around the same time, I was quite intrigued by a study that came out of Philippe Vallet's group in France, where they show that apelin can improve glucose utilization in both normal mice as well as in obese insulin-resistant mice. There was a lot of kind of hype around it. NPR calling this new drug potentially for diabetes. Unfortunately, that was 2008, and we're now in 2019, and yet no one has actually really tested this, I guess, to a great extent in humans with diabetes. There was one study that was published a couple of years ago, which did demonstrate some human efficacy data where apelin injection, this is injection of the peptide, which was found to lead to some degree of improved insulin sensitivity in overweight men when these humans were undergoing a hyperinsulinemic euglycemic clamp. So there's some suggestion that this pathway might have a benefit in human diabetic patients, but to date there has been no long-term study to test this effect in human diabetes or human atherosclerosis. But what got me kind of intrigued based on what what we and others have found about this apelin pathway is, it does seem to have this robust glucose utilization effect. It does seem to have a fairly induced improved insulin sensitivity. But the key questions that kind of I thought needed to be addressed were what cells express this receptor, because if think, any cell, whichever cell is the highest expression of the receptor, or where the receptor is expressed, is likely to be having the key role in regulating downstream effects, including the metabolic effects. And the second question in conjunction with that is, what is the mechanism of these downstream effects of apelin APJ pathway in the metabolic side? So that was kind of our study that I'll be focusing on for the rest of the talk today. That was published about a couple of years ago. We have other work in the field in looking at endothelial regulation and metabolism, but some of that work is currently in preparation and should be hopefully coming out shortly. So what we found, and with help of Christy Redhorse, who was a collaborator on this project, and who, by the way, is a speaker in next month's NAVBO webinar, was that this receptor is extremely highly expressed in the endothelial cells. So we took both human tissues and did in situ hybridization to look at APJ, and found that the APJ receptor was predominantly expressed in these microvascular endothelial cells. We also used this APJ Cree reporter mouse crossed to the Rosa MTMG which ends up labeling every cell that expresses APJ or apelin receptor green, and found that majority of APJ, as you can see here in green, in skeletal muscle, white adipose, and brown adipose, was predominantly restricted to the endothelial cells, which are labeled red here with VE cadherin staining. So based on these two studies, and when we isolate the endothelial cells, the isolated endothelial cells had a much higher expression of APJ compared to total tissue homogeneous, as shown here for skeletal muscle, as well as brown adipose tissue. So these findings really confirmed in our minds that most of the APJ expression is in the endothelial cells and not in other cell types. And this is the work of Hyung and Jingxiu Wu, who are two postdocs who spearheaded this project. So we were trying to make sense of how could this endothelial-based pathway really regulate this whole body metabolism, and we turned to the literature and what had been published before with regards to various other endothelial mechanisms that may be playing a role in regulating glucose utilization and insulin sensitivity. Work from Ulf Eriksson's group over the years have implicated that VEGF-B might be important in regulating this trans-endothelial fatty acid uptake. Jorge Plutsky had also published that endothelial PPAR gamma might be involved in regulating this trans-endothelial fatty acid uptake that ends up regulating insulin sensitivity and glucose utilization. Zoltarani's group had published more recently that 3-hydroxyisobutyrate might be involved in a similar manner of this trans-endothelial fatty acid transport. So the concept here is that endothelial cells can regulate how much fatty acid can cross the endothelial barrier, which ultimately affects how the whole body responds to glucose and insulin. And this is a concept that's been around for many years, that excess adipose or excess fatty acid buildup in tissues such as skeletal muscle will lead to ultimately impaired insulin signaling and impaired glucose utilization, which would be kind of two of the hallmarks of diabetes. So we were curious to know this apelin pathway, which also is predominantly endothelial based on our expression profiling. Could that also have a similar effect in regulating this trans-endothelial fatty acid transport? So we started out with a number of in vitro studies where we basically plated out a layer of endothelial cells in a trans wall, and then added fluorescently labeled BODIP to the top, of fluorescent labeled fatty acid, BODIP to the top, and then basically quantitated the amount of fluorescent fatty acid that's transported across the endothelial layer. And we found that when these cells were pre-treated with apelin, there was a significant reduction in the transendothelial fatty acid transport. On the contrary, when we knocked down APJ, the receptor, we found that there was a significant increase in this transendothelial fatty acid transport. We wanted to see if we could potentially test this in vivo as well. So we took the apelin knockout mouse. This is the ligand knockout mouse, and fed them oral gavage of radioactive oleic acid, C14 oleic acid, and measured how much of the fatty acid gets taken up into various organs. And we found that the apelin knockout mice had a significantly increased uptake of fat in organs such as liver, brown adipose, heart to some degree, as well as skeletal muscle. These are two femoral muscle and dorsalis muscle, where there was a significant increase in the fatty acid accumulation in the knockout compared to controls. Again, and then the two data sets supporting each other, that endothelial apelin seems to be a key regulator of transendothelial fatty acid transport. So to really try to hammer home the point that this might be an endothelial APJ or apelin receptor driven mechanism, we generated endothelial specific APJ knockout mouse. These are using Ralph Adams inducible V-cadherin Cree, where the APJ deletion was induced in adult mice. And then we conducted glucose tolerance testing, where we give a bolus of glucose and then measure the glucose levels thereafter. We found that compared to control mice, here shown in filled circles, the endothelial apelin receptor knockout mice had a much greater level of glucose utilization, after which supported our hypothesis that perhaps this is an endothelial-driven mechanism that's regulating how glucose is utilized in these mice. We also found that these mice actually fail to respond to exogenous apelin. So what had been shown previously in that paper from Cell Metabolism from Philip Vallet's group was when you give mice apelin, a pre-injection with apelin, these mice become much more, utilize glucose much better. So this is a control mouse with just PBS injection. And what you see is in mice that received apelin, you see a much less increase in glucose after the GTT, after the glucose injection in a glucose tolerance testing. What we found in the APJ knockout mouse, these are endothelial specific knockout mice, we found that these mice basically had a higher level of glucose to start with, as shown here, going up to mid 600s versus 400s in the control mice. And they also failed to have any response to apelin, again, supporting the hypothesis that this is an endothelial-based mechanism that might be playing a role. We wanted to further investigate what is the mechanism, what drives downstream signaling by this apelin signaling that might be playing a role here. And a number of studies had implicated that AKT itself might be a downstream target of apelin, but a key target of AKT, namely FOXO1, was really not tested to be a downstream apelin APJ target in endothelial cells. FOXO1 has obviously been implicated to be a key downstream target of AKT in a number of studies by other investigators in the vascular biology field. And FOXO1 also is, in developmental context, certainly it's an important developmental gene, but in the adult kind of homeostatic context, FOXO1 is predominantly thought to be a gene, a transcription factor that might promote endothelial dysfunction in the context of diabetes, atherosclerosis, and other metabolic and vascular disease context. So given that apelin has already been implicated to be regulating AKT, we wanted to see if could apelin also be regulating FOXO1. And we found that when we stimulate endothelial cells with apelin, there was a robust increase in FOXO1 phosphorylation, as seen here, both in a concentration-dependent and time-dependent manner. This induction of FOXO1 phosphorylation also led to cytoplasmic translocation of FOXL1. This is without apelin here, FOXL1 in red versus with apelin, with FOXL1 here moving out to the cytoplasm. So apelin seems to be a key regulator of endothelial FOXL1 activation or inactivation, where apelin actually induces phosphorylation of FOXL1, cytoplasmic translocation, and perhaps ultimately degradation of FOXL1. So given this inverse relationship between apelin, APJ, and FOXL1, we wanted to try to delve into what could be a downstream target of this pathway that might be playing a role in how these endothelial cells can handle fatty acid transport and uptake. So we conducted a gene expression profile analysis to see if there might be perhaps gene products that are differentially regulated, which could potentially explain how perturbation of apelin pathway might lead to this dysregulated fatty acid transfer. And this is basically a ratio plot looking at the genes that are differentially regulated in the context of apelin APJ knockdown and FOXL1 knockdown in HUVECs. And we were very interested to see that FABP4, or fatty acid binding protein 4, was one of the genes that was most highly differentially regulated in this context, and that this gene went up significantly when you knock down apelin and APJ, and it went down dramatically when you knock down FOXL1. And we were able to confirm the array data with Western blot as shown here. When we knock down apelin, you see a robust increase in FABP4 in HUVECs. And when you knock down FOXL1, you see FABP4 significantly decreased. And when you do a double knockdown of both, you actually see a decrease, suggesting that FOXL1 may be downstream of apelin in regulating FABP4 expression. We also corroborated this in vivo, where apelin knockout mice had significantly increased expression of FABP4 in the endothelial cells, and this was confirmed by Western blot as well, shown here. And FOXL1 endothelial specific knockout mice had decreased expression of FABP4 both in endothelial cells by immunohistochemistry as well as by Western blot. So, I'm not saying this is the only target, but FABP4 seemed to be a target that might make sense in terms of how apelin might be regulating this transendothelial fatty acid transport. So we wanted to test that experimentally by taking this, again, going back to the in vitro assay where we played out layer of HUVECs, and we've done this with other endothelial cells as well. And we knock down apelin and APJ, and we see a significant increase in the amount of fatty acid transfer. And when we concurrently treat these cells with an FABP4 inhibitor listed here, we found that this increase in fatty acid transfer could be inhibited or regulated. So this FABP4 does seem to be a key downstream target that's regulating some of the fatty acid transport activity of the apelin APJ pathway. We wanted to see if we could potentially rescue the phenotype of the apelin knockout mouse by targeting these downstream mechanisms. First, we pursued a concurrent deletion of apelin as well as endothelial specific deletion of FOXL1, which led to a significant, and first you see that the apelin knockout mice have significant increase in C14 oleic acid that accumulates in different tissues, including brown adipose, liver, as well as skeletal muscle. And when we generated a double knockout, apelin, FOXL1 endothelial double knockout mice, this increase in fatty acid uptake was abrogated in these mice. And we could also confirm that using oil red O staining of skeletal muscle, where there was much less fat, as shown here by decreased oil red O in the skeletal muscle of the double knockout versus the single knockout mouse. And lastly, we wanted to see, can we also potentially regulate this, demonstrate this, use targeting the FABP4. For the purpose of this study, it would have been also nice to see if we could show this with an FABP4 knockout mice, but those were not available at the time. So we chose to use the the FABP4 inhibitor compound. This is a small molecule compound that appears to be fairly selective for FABP4, may also have some cross-reactivity with FABP5, and wanted to see if this could potentially rescue the apelin knockout phenotype, again, with respect to the fatty acid accumulation. We found that in wild-type mice, this BMS compound, FABP4 inhibitor, had really no effect on fatty acid accumulation, shown here comparing the white and the red bars here. But when the apelin knockout mice, again, these are the mice where there's a significant increase in fatty acid accumulation in the different tissues. So these mice when these mice were treated with the FABP4 inhibitor, we saw that there was a significant reduction in the fatty acid accumulation in the different tissues. Again, further supporting the hypothesis that FABP4 may be a key downstream target of apelin signaling in this context. And lastly, we wanted to see exactly, could we potentially use this compound, this FABP4 inhibitor, to rescue the phenotype of the APJ endothelial specific knockout mouse. Again, to really validate the finding that endothelial pathway, endothelial APJ may be critical to regulating some of these metabolic effects. So what we did initially was to take these APJ endothelial knockout mice, again, these mice were induced to delete APJ in adulthood using the CDH5 tamoxifen-inducible Cree. And these mice were, as shown here, these mice were treated with this FABP4 inhibitor compound, BMS 309403. And we found that when the APJ knockout mice were treated with this compound, again, we saw a significant decrease in the glucose levels after GTT study, as shown here in open dots versus closed circles. And we also found that there was a significant increase in fatty acid accumulation here, shown here in oil red O staining, comparing controls to APJ knockout. And that was also reversed to some degree when these APJ knockout mice were treated with this FABP4 inhibitor BMS compound. So overall, again, these data support the hypothesis that this appears to be a fairly endothelial APJ specific phenomenon, that the endothelial apelin APJ signaling appears to be having a critical role in regulating how glucose is utilized and how fatty acids is transported, and how overall the animal might be responding to glucose and insulin levels. So previous studies, which I didn't have much time to get into, but including the original cell metabolism study, have suggested that perhaps this apelin and the metabolic benefits of apelin may be directly occurring in cells such as myocytes and other downstream target cells. But based on the finding that most of the apelin receptor seems to be in the endothelial cells, what we propose is that this mechanism may be endothelial dependent, in that there is really no direct effect of apelin on the myocytes or cardiomyocytes, but is this all potentially a secondary effect driven by the effect of apelin APJ signaling on the endothelial cells that are quite abundant in many tissues, including skeletal muscle, cardiac tissue, as well as others. And we were pushing forward this hypothesis that this barrier function of endothelial cells may be something that's directly targeted by apelin APJ signaling, which may be playing a critical role in how the whole body ends up utilizing glucose, and how body becomes sensitive to or resistant to insulin. And that may have a key role in the context of human disease, diabetes, prediabetes, and metabolic syndrome. So based on much of the data I presented today and data from a number of other groups, I think apelin does a lot of beneficial things in different contexts. In patients with heart failure, again, I didn't have time to show you that data. There appears to be fairly good protective effect by acute injection of apelin. patients with pulmonary hypertension also seem to have some benefit to apelin infusion in the acute setting. And also diabetes and potentially cardiovascular disease such as atherosclerosis as well. But the main limitation we have to face is that this is a peptide-based most of these studies have been done using a peptide, which as a therapeutic modality would have to be injected. The peptide itself has an extremely short half-life, about 2 to 8 minutes in serum. And ultimately, just using an activator or peptide ligand may lead to desensitization of the receptor, which may end up losing efficacy in the long run. So we're making some efforts to try to bypass some of these limitations, either through trying to develop small molecule compounds that activate the APJ receptors, or working with compounds that have been made by other groups to see if those compounds might have a similar kind of an effect. We've tested one of these APJ agonists, a small molecule, and we have found, fortunately, that the agonist appears to have a similar effect as the apelin peptide itself. Again, this is a fairly early pilot study, so more studies will need to be done, but basically the hope is that perhaps a longer acting form of this APJ activator, maybe a small molecule or modified peptide, might provide us with a better capacity to really engage this pathway as a potential drug therapy in different cardiovascular or metabolic disease contexts. So that's some of the work that's been ongoing in my lab. A number of drug companies are also pursuing APJ activators, and we're looking forward to see how some of those studies pan out. So just to start to conclude, we feel that activation of this apelin APJ pathway promotes various aspects of atheroprotection as well as metabolic homeostatic effects that might be very synergistic with one another. And we found that a lot of the metabolic effects of apelin may be, at least in large part, due to the endothelial basis of this apelin APJ signaling, given the robust data we have observed in our conditional endothelial specific APJ knockout mice. And just to that point, this APJ endothelial knockout mice have significantly impaired glucose utilization and impaired response to apelin, which could explain a lot of the phenotype that's been found in response to apelin injection in some of the earlier metabolic studies. And apelin, a key target of apelin appears to be this endothelial FABP4, which is a key regulator of fatty acid uptake and transfer across the endothelial layer. And lastly, we are hoping that if we can activate this or engage this apelin pathway, that may be a viable therapeutic strategy in diabetes, and which may ultimately be a pathway similar to empagliflozin, similar to some of the GLP-1 pathway targets. Could we potentially target this pathway to achieve improved diabetes care as well as improve cardiovascular benefits? That's hope at this point, something that hopefully will be tested sometime in the near future. So with that, I'll conclude by thanking everybody involved. Most of the work I showed was Hyung and Jingze Wu, who carried out the work in looking at the endothelial APJ metabolic studies. A number of other members of my lab, current and former members of the lab, as well as multiple collaborators who's contributed to this study. Picture of our recent lab here, and some of the funding sources down at the bottom. And I'll finish off by giving a plug for Christy Redhorse. Again, key collaborator on some of our studies will be our presenter next month. And I'll finish there and happy to take any questions.
Thank you so much, Dr. Chun. It was a fantastic talk. I have a few questions, and then I'll yield the turn for Dr. Clapham, then she can ask the questions of the audience. One of them is, at the beginning of your talk, you mentioned that there's 2 ligands for the APJ. One was apelin, the other one was elabella, I think you mentioned. Does it function like apelin? Does it have also potential therapeutics?
Yeah, no, that's a great question. So there have been some studies in which the effects of the two peptides have been compared side to side. Some of these studies are studies by Anthony Davenport looking in the role in the context of pulmonary hypertension, where they seem to be behaving fairly similarly in improving that particular disease context. Now, other studies, including a number of elegant studies by Bruno Riverside at Singapore, A-STAR, have demonstrated some unique roles for these two particular ligands in the developmental context. In the metabolic context, we have done some initial studies looking to see what the response would be in response to injection of a Elabella peptide, and there is an improvement in the GTT study, but we haven't quite teased out exactly how different or similar the two peptides might be.
Thank you so much. So at this point, I'm gonna yield to Dr. Clapham, and she's gonna ask you some of the questions of the audience. Thank you.
Okay, thank you. So the first question from the audience is, are there any human diseases known to be associated with a mutation in apelin?
Yeah, great question. I mean, I wish there was, but so far there's nothing that's been conclusive. I mean, there's been some genetic variants which might be more prevalent in specific populations with hypertension, perhaps some stroke signal, but again, I don't think any of these studies are really conclusive in demonstrating a role in a cardiovascular metabolic disease context. I mean, it's possible that the mutations might be lethal, but again, or maybe we haven't looked hard enough. But and I'd love to see a signal there at sometime in the near future.
The next question is, what is known about apelin expression in tumor-associated endothelial cells?
Yeah, that's a great question. I don't think I have a good answer to that. I mean, I think there have been studies where groups have looked at perhaps neutralizing apelin as a tumor therapy, and some of these studies actually showed some promise. And I think some companies might have formed around that idea that you can neutralize apelin with a neutralizing antibody and perhaps inhibit tumor growth. But in terms of expression of the ligand or the receptor in the tumor context, I'm not 100% sure that's been well defined, or I just don't have a good answer.
Okay. The next question is, does endothelial specific apelin knockout show hemorrhage and edema in adults? I guess adult mice.
Yeah. So the endothelial specific ligand knockout, as far as I know, I think that was generated by a Japanese group a few years back. I don't believe they had an overt phenotype. So in our hands, our global apelin knockouts don't have any adult phenotype. There is some delayed vascular maturation, but there does not seem to be, once these animals grow to be adults, there's really no major phenotype. APJ knockout, they have a more severe developmental phenotype, but again, some of those animals can survive into adulthood. And overall, there does not seem to be an overt hemorrhage phenotype in adult mice. And even in our conditional knockout mice, we didn't observe any long-term effects of having of deleting APJ in the endothelium in adult mice.
And so far, I have one remaining question from the audience, which is, does the perivascular fat tissue release apelin?
Right. So, I think a lot of the studies have in the beginning, in the earlier days, I think thought, when you basically homogenize adipose and do PCR for apelin, you have a pretty robust signal. But when we've done some of our studies using an apelin LAC-Z reporter mouse, predominantly most of the apelin seems to be coming from the endothelial cells. So, a lot of the earlier studies and a lot of the studies in the literature suggest that apelin is an adipokine coming from adipocytes. But, I'd like to kind of challenge that dogma, because most of the studies that we've done, looking specifically at the apelin transcript, it does seem that apelin is coming predominantly from the endothelial cells, whether it's adipose tissue, cardiac cells, or other organs.
Well, Dr. Cuervo, that's the last question I have from the audience that was written in. I don't know if there's any.
Thank you very much. If anyone still has any questions, they can ask it live by clicking on the hand icon, and that way you can raise your hand and I'll call your name and you can ask your question live. Any questions out there? No? Well, then I'd like to thank you both. If you have any question and you just didn't want to answer it publicly or ask it publicly, please email info@navbo.org and we'll make sure to share it with Dr. Chun. I hope you found this research presentation beneficial. These webinars are brought to you by the NAVBO Education Committee. Our next webinar, as Dr. Chun mentioned, is on Thursday, December 19th from 1 to 2 p.m. Eastern Standard Time. And we will have Christy Redhorse from Stanford University presenting a webinar entitled Coronary Blood Vessels Play Multiple Roles in Cardiac Development. Look for the registration information in the NAVBO News Beat newsletter. And please fill out the GoToWebinar evaluation form when you receive it in a subsequent email. And let us know what you think of the GoToWebinar format and any topics you may want to see in a future NAVBO webinar. So thank you everyone for attending. Thank you, Dr. Chun. Have a good day.
Thanks, everybody.